Okay, for Pooja, good evening. So today we're going to start our fourth lecture, and the fourth lecture is fully based on membrane transport. I hope in the previous lecture we saw about the various types of connection that is existing between the plasma membrane and how they take up the role in transport of molecules. And today's topic actually having five important areas to be discussed. So introduction is over. We have a diffusion. In that we have facilitated diffusion for ionic and membrane transporters, active transport using sodium potassium pump and other ion pumps, and secondary active transport, and the importance of membrane transport, and we have a small summary to be discussed about that. In that topic today we'll be focusing only on diffusion. So it is a lecture that you have requested since you're physically not available in the online. So I'm recording and sending it to you. So any doubt is there in the next class, we can discuss about that. So membrane permeability. That means the plasma membrane is actually a permeable membrane. But what type of permeability do they exhibit? Are they selectively permeable or they are freely permeable? So that becomes a question. So permeability is an important concept for the plasma membrane. It will decide what type of molecule has to be transported and where the molecule has to be transported, whether all the molecules can I allow or selected molecules can I allow. So based on the requirement of a cell, plasma membrane is actually mostly it's a semi-permeable membrane. It's not a freely permeable membrane. The semi-permeable membrane. At the same time, it is also a selectively permeable membrane. So regarding the functions of plasma membrane, the main thing we're going to focus on receptors, but already we have done about that. The topic will be mostly focused on transport. So when we look about transport, so selective permeability of molecules. That is, the small molecules or outer membrane is completely hydrophobic in nature, and the inner one is completely hydrophobic. Okay, about when you look about the structure of plasma membrane, let me recall here. So this is your glycerol and your fatty acids are there here. This end is completely hydrophilic in nature, whereas here it is hydrophobic in nature. Because of that, small molecules or some of the hydrophobic molecules they can easily pass through by diffusion. But there are some ion molecules are there which are not at all be able to be transported. So for that we require some transporter protein. So regarding the transport system in case of plasma membrane, there are two types of transport. One is passive transport and the second one is active transport. So we have two means by which the molecule can be transported. So for example, let us take as this is the plasma membrane. So I've taken a plasma membrane over here. Let me take eraser and separate between these two. The work is done. Now regarding the movement of molecules, we're going to focus. In passive transport, okay, these are the molecules which has to be transported by the plasma membrane. Here, the molecular concentration is very high. In this case, molecular concentration is very low. So the molecules are able to move from higher concentration to a region of lower concentration. When a molecule moves from a region of higher concentration to a region of lower concentration, it is called downhill movement where no energy is utilized. This type of transport is called as passive transport. At the same time, if the molecules are able to move from if the molecules are able to move from lower concentration to a region of higher concentration. The molecules are able to move from a region of lower concentration to a region of higher concentration. So the molecules are able to go against the concentration gradient. Since they're able to go against the concentration gradient from lower concentration to a region of higher concentration, these molecules utilize energy in the form of ATP. So ATP is broken down into adenosine diphosphate and another molecule of inorganic phosphate is utilized for the transport of molecules. So here the molecules are going along the concentration gradient. So it is called as downhill movement. And since they're going along the concentration gradient, there's no ATP utilization. There's no ATP utilization. This is called as passive transport. Here, the molecules are going against the concentration gradient. Since the molecules are able to go against the concentration gradient, it is called as an uphill movement. So your energy in the form of ATP is completely utilized. This type of transport is called as active transport. So in passive transport, we'll be discussing about diffusion, an important phenomena which is always occurring in a organism, that's called as diffusion, followed by osmosis. In active transport, only one transport energy is utilized in the form of ATP. So let us focus on diffusion. Diffusion is a means of transport where molecules are able to move from region of higher concentration to region of lower concentration, or an higher electrochemical potential to region of lower electrochemical potential. So the movements are random. So the molecules are able to move uniformly from a region of higher concentration to a region of lower concentration. What is diffusion? You could have learned in your grade 9, where copper sulfate, or you can say potassium permanganate, they could have kept in a beaker. Slowly they could have diffused. Or agarbatti, when you place in a corner of your room, it will spread everywhere. That is because of diffusion. So it is always going to occur along the concentration gradient. Now, when look about this passive transport, I'm going to go ahead with the passive transport where no energy is utilized. In diffusion, there are two types of diffusion. It's a general diffusion, or commonly called as diffusion, where we have next group of diffusion that is called as facilitated diffusion. So we have two types of diffusion. It's a general diffusion or facilitated diffusion. How to understand this concept? Okay, very simple. Let us draw again the plasma membrane. And you know that the molecules are able to move from a region of higher concentration or higher electrochemical potential to a region of lower electrochemical potential. So no change here. The molecules are there in higher level. Here also the molecules are there in higher level. Here the molecules are there in lower level. Here the molecules are there in lower level. Without any carrier protein, if the molecules are able to move from higher concentration to a region of lower concentration without any membrane protein, from higher concentration to lower concentration, it is called as general diffusion. At the same time, if we have some selected molecules, because I told you the plasma membrane is able to transfer only by selected molecules. So when the plasma membrane able to transport the molecule in selectively permeable way, that means what? In that cases, I'll just make a small mark here where I'm going to go draw some carrier protein. I told you the importance of carrier protein. So means of some carrier protein, that is or channel protein, the molecules are transported without any utilization of energy. It is a channel protein, or it is also a tunnel protein, or it is also a transport protein. Lot of names are there for it. They name for it. So using these molecules, using these channel protein, if the molecules are able to move from higher concentration to a region of lower concentration, This mechanism is called as facilitated diffusion. Now, this facilitated diffusion is of three different types. So, let me draw only the facilitated diffusion now. So, let me take one eraser. Okay, let me draw the membrane proteins. Let me extend these lines. Now, in the first case, okay, let me draw the channel proteins over here. These are the channel proteins. Okay, recording is on process only, so nothing to worry about that. Only thing is in the Google Classroom, it is not a joint. That's not a big deal. I think recording is on process. Let me pass it. Yeah, the video is resumed now. So we have a set of channel proteins here. So we got a set of channel protein. This channel protein helps in transport of molecules, which already have told you that is they are helpful in the transport of molecules. Now, when you look about these channel proteins, if the molecules are able to move only in one direction, ஒரே ஒரு பக்கம் தான் move ஆகுது. That is only in one direction. If two molecules are able to go simultaneously in the same direction, that is about the channel proteins, or they allow the movement of molecules in opposite directions. So three different pathway mechanisms are there for facilitated. In general one, we have only one means of transport. Here what I've explained to you is another facilitated. Here the molecules are able to be transported only in one specific direction. This is called as uniport facilitated diffusion or uniport pathway of diffusion. Uniport pathway of diffusion. If the molecules are able to go through simultaneously, maybe input or output, it's fine. But they're able to go only in the same direction, means it is called as symport mechanism. But if they go up in simultaneously in both the direction, that is the aquaporins or the transport protein, can carry the molecules in both the direction, both input and output or inflow and outflow. This is called as antiport mechanism. This mechanism is called as antiport mechanism. So you have for these are the following types of passive transport. We have osmosis also that we can discuss little bit later. I think today I won't be discussing osmosis to you also. So simple way to understand diffusion puja in passive process. Let me take a small example. See, I'm going to take one beaker for your understanding. I'm taking this. I'm just taking one beaker. Let me fill this beaker with completely with water. So we have filled the beaker completely with the water. Okay, let us just go with only few levels of water. I have filled this beaker with water. I am taking some a drop of copper sulfate. Let me take a copper sulfate here. After some time, what I can see is in random movement, this copper sulfate move in various direction and they get uniformly distributed in later phase. Because in this water molecule, we don't have any copper sulfate. Only the corner we are placed. Slowly, these molecules will be diffusing. So from a region of higher concentration to a region of lower concentration of copper sulfate. This is called diffusion. Where some proteins are used up means it is called as facilitated. If no proteins are utilized, that is called as general or basic diffusion process. So that is what I'm going to teach you here, that how the diffusion of these molecules will take place. So the membrane transport, I'm going to discuss about the passive transport. In passive transport, there's no utilization of energy. The molecules are able to move from higher concentration to a region of lower concentration. In passive and simple diffusion, we don't require any transport protein, but in other like facilitated diffusion, we require actually a channel protein or a transporter protein for transport of molecules. But in passive transport, the molecule, or in active transport, the molecules are going from higher or lower concentration to higher concentration against the concentration gradient. So they utilize energy in the form of ATP. I hope you're able to understand. Since you're not there with us physically, so I won't be able to know that whether you have understood. That's why even recording, I'm just keeping the time as minimum instead of 1 hour. I'm going to keep it as nearly 35 or 30 to 40 minutes, so that you'll be able to understand what actually I mean for you to understand. Next. Simple diffusion, I told you. So, simple diffusion, the molecules are able to move from higher concentration to a region of lower concentration. So, it is along the concentration gradient. The molecules are able to go along with the concentration gradient. So, this is the extracellular fluid and this is the intracellular fluid. We're going to call as ECF, extracellular fluid. This is called ICF, it is called intracellular fluid. So what we can observe is the molecules always goes from a higher concentration to a region of lower concentration. Simple diffusion, there is no carrier protein at all. As a time proceeds, you can say the molecule could have been more used up. So most of the molecules are freely permitted inside the cell. This is called simple diffusion. So simple diffusion you can understand with respect to the osmosis too. So what is osmosis? I'm going to introduce that about osmosis now with the help of simple diffusion. So the concept I'm taking is osmosis. Osmosis is movement of solvent molecules. It will not talk about solute molecules. It talks about movement of solvent molecules. Solvent in the sense, if I take sugar, if I take water, if I mix them each other. But today osmosis definition will be taught for you, and with that I'll be stopping for you. So it will form sugar solution. So sugar is actually a solute. Solvent is actually a water. So osmosis says about where the solvent molecules are able to move, water molecules. The water molecule moves from higher concentration to a region of lower concentration. So simple terms I can define as osmosis is movement of molecules from a region of higher concentration to a region of lower concentration through semi-permeable membrane. Now, a small change I'm going to introduce in the definition. Instead of movement of molecules, solvent molecules, which is actually water. தண்ணி மட்டும் தான் மூவ் ஆகும் sugar from a region of higher concentration to a region of lower concentration through a semi-permeable membrane is called as osmosis. So, what is semi-permeable membrane? The membrane that allows only the solvent molecules to pass through is called as a semi-permeable membrane. So, இங்க ரெண்டு விதமான solution இருக்கு. The term I will be using a little bit later, hypertonic and hypotonic. The first condition, இங்க பாருங்க, water concentration I'm taking here. Water concentration is high here. Solute concentration is low when we compare the two medium. Say this is a medium எடுத்துக்கலாம், this is the B medium எடுத்துக்கலாம். When water concentration is high, solute concentration is very low. So since solute is very low, it is called a tonic. Tonic stands for solute. It is hypotonic solution. It is called as hypotonic solution. At the same time இங்க பாத்தீங்கன்னா compared to the cell A, in this water concentration is comparatively less. Solute concentration, which is given in purple color, is more. So the molecules goes from higher concentration to lower concentration. But cell A la water molecules அதிகமா இருக்கு because solvent molecules are very high. In this one, solid concentration is more. So osmosis says that water molecules moves from higher concentration to a region of lower concentration. So here water concentration is very high, here water concentration is very low. As a result of this, the molecules are able to go from higher concentration to lower concentration. So molecules goes from cell A to cell B. So cell B la concentration 1, 2, 3, 4, 5, 6. Inge 1, 2, 3, 4, 5, 6. Inge 6 molecule 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. No change in the solute. இங்கேயும் எனக்கு கம்பேர் பண்ணி பார்த்தீங்கன்னா 1 2 3 4 5 6 7 8 9 10 11 12 13 1 2 3 4 5 6 7 8 9 10 11 12 13. So the concentration of molecules are remaining the same. That is the solid molecules. Only water concentration alone increased in cell B because from hypotonic they went to hypertonic. So there are three types of solution to be discussed about osmosis. One is called as hypotonic solution, hyper அதிகமா hypertonic solution. Next one is isotonic solution. Three types of solution are there. One is hypotonic, hypertonic and isotonic. So three types of solutions are there. Hypotonic means water concentration is very, or solvent concentration is very high compared to the another cell, and solute concentration low. Hypertonic, tonic stands for solute. So hypertonic means the concentration of solute is more, water is less. Isotonic means more or less a balanced solution. So the water molecules moves from less concentrated. That means what? More, or you can say higher water, from a higher water concentration to region of low water concentration through an important membrane called SPM, semi-permeable membrane, that is called osmosis. So this is a simple solution. You can see what will occur to the RBC. I'm taking one RBC. RBC when I'm dipping in a hypotonic solution naka, hypo naka thanni concentration very irukku. So I have a RBC. This I'm dipping in hypotonic solution. Hypotonic la water concentration is very high. So the water will enter into the cell by a process called as osmosis. What osmosis means? Endosmosis. That is endoosmosis or endosmosis. When I drop them in a hyper salt water, RBC இருக்கிற தண்ணி ஃபுல்லா வெளியே போயிடும். It is called as exosmosis. The RBC will shrink. The RBC will swell and will burst. ஏன் burst ஆகும்னா cell wall இருக்கா? That's a different one. When I drop an isotonic solution, there will be no changes. ஒரு change நடக்காது because it is a balanced solution. So we have isotonic medium where equal concentration of solute is maintained, so no water movement. Hypertonic is a normal water, just put some resin, it will swell in case of plant cell. Animal cell will burst because of hemolysis. And that cell is called turgid cell and will break. In hypertonic, the cell will undergo plasmolysis, it will shrink. So today what we have focused is, today we have focused on the membrane transport, why membrane transport are very important, how the selected molecules are able to pass through, what is active and what is passive transport, which is called uphill movement, which is called downhill movement, where it will go along the concentration gradient, where it will go against the concentration gradient. What are the two types of passive transport? One is diffusion and facilitated, and types of facilitated diffusion based on the porins as uniport, symport, and antiports. Based on what direction the molecules are able to go from higher concentration to a region of lower concentration. So this is what we discussed: active and passive differences, and how the molecules are able to go in diffusion with respect to osmosis. where water molecules are able to move from higher concentration to a region of lower concentration. Solutes will not move in semi-permeable membrane. Water molecules can pass through. And we have seen the behavior of an animal cell when we dip in isotonic and hypotonic followed by hypertonic. Isotonic, no changes. Hypotonic, the cell will take in the water by endoosmosis and the cell will burst because of absence of cell wall in case of animal cell. But in case of plant cell, it will not burst. In hypertonic solution, the cell will shrink. That condition is called plasmolysis. So, facilitated, I have discussed you about channel protein and transporter protein, which helps in transport of molecules along the concentration gradient. So with this, I'll stop it for today and I'll stop your